welcome to Brain Binge. We're going to start today with a paradox, one that's at the very heart of your existence.
It's a big one.
Every single cell in your body, from the neuron that's processing this very sentence to the liver cell managing your metabolism, they all contain almost the exact same complete set of genetic instructions.
And yet they are completely different. They look different, they act different, their identities are stable, predictable, and totally unique.
That really is the central mystery of multicellular life, isn't it?
Yeah.
How does that genetic uniformity lead to this just staggering functional diversity?
Exactly. If the DNA sequence itself is the fixed instruction manual, the blueprint that never changes, then the really essential question for biology becomes, what is managing those instructions?
What's deciding which pages of the book are open and which ones are sealed shut forever?
That's it. And that is the realm we're exploring today. We're going into that hidden operating system, the layered control architecture that dictates how your genetic code gets used without ever altering the code itself.
We're talking about the field of epigenetics.
And if this precise, elegant system defines who you are, what happens when it fails? If that system degrades over time, and that's what drives aging,
then the big question is, can you fix it? Is it possible to rewind that clock, not by changing the DNA, but by somehow restoring the factory settings of the software that manages it?
That is the multi-billion-dollar question.
So our mission in this Brain Binge is to explore these molecular switches that define your cells, trace how they respond to the world around you, everything from your diet to your sleep, and then really examine the revolutionary but also very risky potential of what's called cellular reprogramming.
And what all of this means for our understanding of age, health, and of course, disease.
Okay, so to set the stage properly, let's start with a really clear core definition. What exactly is epigenetics?
Put simply, it's any stable, heritable, and often reversible change in gene activity that happens outside of the DNA sequence itself.
It's the manager's the blueprint, not the blueprint.
Precisely. That analogy of the instruction manual is just so crucial here. The DNA, the A, T, C, and GS, that's the manual. It's fixed, it's complete, and it's in every single room of the house.
But the epigenetic system is everything else. It's the sticky notes, the highlighters, the chapters that are paperclip shut.
It's the librarian who decides which departments even get to look at the book. The DNA sets the potential, everything the cell could be. But gene expression, whether a gene is on or off, is intensely dynamic.
Yet epigenetics is what controls that dynamism.
It's what allows a cell with the potential to be anything to commit completely and stably to being just a muscle cell or just a neuron for its entire life.
So if the DNA is already fixed, why did life evolve this incredibly complicated second layer of control? Why not just hardwire everything into the genes from the start?
Because multicellular organisms have needs that pure genetic determinism just can't meet. The first is what we just talked about, cellular specialization.
Differentiation.
Right. The second is long-term memory. How does a liver cell, which divides all the time, remember after thousands of divisions that it has to stay a liver cell? That memory isn't in the DNA sequence, it's imposed by the epigenetic state.
And the third requirement must be flexibility, the ability to adapt.
Absolutely. Environmental flexibility is the masterpiece of the epigenetic system. It allows an organism to fine-tune its gene expression in response to the world.
Things like stress, nutrition.
Exactly. Stress, nutrition, even the daily circadian rhythm. Without this high-level control, our cellular identity would be fragile, development would be unstable. We just couldn't adapt to a changing world.
And this idea that there's a regulatory system above the genes, it's actually quite old, isn't it? Even though the molecular details are pretty modern.
Oh, it's much older than most people think. We have to credit the biologist Conrad Waddington. This was back in the early 1940s, before we even knew the structure of DNA. He coined the term epigenetics.
But he wasn't talking about molecules and chemical tags.
No, not at all. He was talking about the process of development, and he gave us one of the most powerful metaphors in all of biology, the epigenetic landscape.
It's such a great mental image. Can you walk us through that?
Okay, so imagine a ball sitting at the top of a vast, sloped terrain. That ball is an embryonic stem cell. It has the potential to roll in any direction. Total potential. But as it starts to roll downhill, the terrain isn't smooth. It's carved with channels, with deep ravines and valleys. Each valley is a specific developmental path. One leads to muscle, another to bone, another to brain tissue.
And once that ball settles deep into a valley, once the cell is fully differentiated, it's incredibly hard to push it back up the hill or over into a different valley.
Exactly. The shape of that landscape, the depth of those valleys, is sculpted by the epigenetic system. It determines the cell's fate, and the stability of the valley ensures that identity is locked in for good. It was genius, really, to see that development was a constrained path, not just a passive reading of DNA.
But for a long time, this idea was sort of sidelined.
It was. The mid-20th century was the explosion of molecular genetics. The double helix, the genetic code, DNA makes RNA makes protein. The focus became almost entirely on the sequence.
Right, all about the base pairs, mutations, the fixed text.
And for decades, epigenetics was often treated like noise, a kind of static that didn't really matter as much as the pure genetic signal. It wasn't until the 70s, and then really the 90s, when we developed the tools to see the molecules that we confirmed Waddington's vision.
You realized it wasn't secondary at all.
It was the core regulatory architecture, and that really set the stage for everything we understand today.
Okay, so if the epigenome is the manager, let's get into the specifics. What are the molecular tools it uses to enforce all this? The first thing to deal with is just the physical reality of packaging.
That's the foundation of it all. I mean, think about it. Every single one of your cells has to store nearly 6 feet of DNA inside a microscopic nucleus.
It can't just be a tangled mess.
Exactly. It's compacted into this organized, dynamic structure called chromatin. That's the DNA wrapped tightly around spool-like proteins called histones.
And that physical structure is the first layer of control.
It is the basis of control. If that DNA is wound up super tightly, we call that heterochromatin. It's silent. It's inaccessible. The machinery literally can't get to it to read the genes.
But if it's loosened up and open?
That's euchromatin. And that's active and ready for gene expression. So the epigenetic machinery is constantly working to manipulate that packaging.
So let's talk about the first major tool it uses, DNA methylation. This seems like the most stable, longest-lasting mark.
It's the chemical anchor, for sure. It's a very simple process, chemically speaking. You're just adding a tiny methyl group 1 carbon, 3 hydrogens directly onto one of the DNA bases.
Specifically onto the cytosine bases.
Almost exclusively on cytosines, and usually when they're followed by a guanine, we call those CPG sites.
And what does sticking that methyl tag on a cytosine actually do?
It's a brake pedal, a very strong one. Methylation is overwhelmingly associated with gene silencing. It physically blocks the sites where activating proteins would bind, and it also recruits other proteins that help compact the chromatin even further.
So it's a multi-layered off switch.
A very robust one. It's central to things like silencing one of the 2 X chromosomes in females. It's a permanent decision.
And because it's a direct modification to the DNA itself, it's incredibly stable through cell division.
And that stability requires some specialized management. There are 2 families of enzymes that do this. First, you have the de novo methyltransferases, DNMT3A and 3B. They're the writers. They put brand new marks down during early development to establish a cell's identity.
And then you have to maintain those marks.
You have to. If a cell divides and the daughter cell loses those marks, the wrong genes could switch on. That would be chaos. So that's where DNMT1, the maintenance methyltransferase, comes in.
How does it work?
When DNA replicates, the new strand is unmethylated. DNMT1 comes along, recognizes the methylation pattern on the old template strand, and faithfully copies that exact same pattern onto the new strand.
It's like a photocopier for the epigenetic marks.
A perfect analogy. It ensures the silencing pattern is inherited perfectly and that the cell lineage stays stable for decades.
Okay, so if DNA methylation is the permanent break, the second mechanism, histone modifications, seems a lot more dynamic.
It is. The histones, those protein spools the DNA wraps around, they have these little tails that stick out, and those tails can be chemically altered in dozens of ways. Acetylation, methylation, phosphorylation. It's a whole language.
Let's talk about acetylation. That one comes up a lot.
Acetylation is a great example of a structural adjuster. Histones have a positive charge, which helps them grip the negatively charged DNA backbone very tightly.
Like a magnet.
Right. Adding an acetyl group neutralizes some of that positive charge. It makes the histone loosen its grip.
So the DNA can relax a little bit, opening up.
And that makes the underlying genes accessible to be read. So histone acetylation is almost always a sign of an active on gene. And removing those acetyl groups, which is done by enzymes called HDACs, tightens things back up.
But histone methylation is way more complex. It can mean on or off.
It's what's often called the histone code, and it's not a simple switch. It all depends on which amino acid on the histone tail gets the methyl group and how many methyl groups are added.
Can you get an example?
Sure. Adding 3 methyl groups to a specific spot, histone 3, lysine 9, is a classic mark for permanently silent chromatin, a lockdown region. But adding 3 methyl groups to a different spot, histone 3, lysine 4, is a signature of an active gene promoter that's ready to go.
So it's a very sophisticated, context-dependent language.
It's a complex language, and the cell has reader proteins that recognize these specific combinations of marks and then act accordingly.
So we have the tags on the DNA, the adjustments to the spools, but if something is really tightly packed, you need actual force to open it.
And that brings us to the heavy machinery, the chromatin remodeling complexes.
The movers and shakers.
Literally. These are huge protein machines that burn ATP, the cell's fuel, to physically slide, evict, or restructure the nucleosomes. They don't just wait for the chemical tags, they use force to push the spools along the DNA.
They're the ones physically prying open the book to the right page.
Prying it open and also snapping it shut. And they're absolutely critical for things like DNA repair. If there's a break in the DNA, the remodelers have to rush in, move the histones out of the way so the repair crew can get access, and then put everything back exactly as it was.
Which means if those remodelers break down, the whole system is in trouble.
Deep trouble. And we know this because mutations in these remodeling complexes are strongly linked to cancer and severe developmental disorders. If the machine that manages the architecture breaks, the whole system can collapse.
And just to add one more layer of complexity, there are also non-coding RNAs involved.
Right. Not all RNA is a blueprint for a protein. Some of it is regulatory. These non-coding RNAs can act like little guide molecules.
How so?
They can bind to the chromatin-modifying complexes, the writers, the erasers, the remodelers, and physically guide them to the right spot on the genome. It adds another layer of precision and stability to the whole system.
It's an incredibly intricate system of checks and balances.
Designed to be both rock-solid, stable, and, when needed, subtly adaptable.
So now that we have the machinery, let's look at what it does in practice. First, that enforcement of cellular identity and memory.
Right. So we start with embryonic cells, which are epigenetically permissive. They're a blank slate, lots of open options.
But as they differentiate,
they get very restrictive. They aggressively silence all the genes for the cell types they're not going to be, and they reinforce the programs for the identity they've chosen.
That's the ball rolling deep into Waddington's Valley.
But you're saying that identity isn't just passively held onto, it has to be actively maintained. Absolutely. This is a point people often miss. Every single time a cell divides, that whole intricate pattern of methyl marks and histone modifications has to be copied perfectly.
And if it's not?
If that maintenance fails, the cell starts to suffer from a kind of amnesia. It forgets what it is. Genes that should be silent might flicker on, and that's a hallmark of functional decline.
So the stability of your tissues depends on the fidelity of this copying process, moment to moment. But the epigenome is also listening to the outside world. How does the environment actually talk to these molecular switches?
It's a really direct link through metabolism. The enzymes that add methyl groups, for example, they need chemical building blocks, cofactors that come from our diet, things like folate and methionine.
So what you eat literally provides the ink for writing the epigenetic code.
Precisely. If you're nutritionally stressed, the availability of that ink changes, and that can disrupt the whole system. And then there's stress. The hormone cortisol is a powerful modifier.
How does a stress hormone translate into an epigenetic mark?
When cortisol is released, its receptors go into the nucleus and bind to DNA, and they recruit chromatin-modifying enzymes. This can cause very rapid changes, especially to histone acetylation at stress response genes.
So it fine-tunes your response to the stressor.
Yes, but if the stress is chronic, especially early in life, those changes can become stable. They can leave long-lasting signatures that affect how you respond to stress decades later.
Let's focus on the brain, because it's such a paradox. It needs incredible stability for a lifetime, but also the plasticity to learn every day.
The nervous system is a beautiful case study. Learning and memory actually involve localized, temporary epigenetic changes. Neurons have to loosen their chromatin just a little bit to turn on new genes to form new synaptic connections.
It's a controlled dance of opening and closing.
It is, but as you mentioned, severe stress can leave a much deeper, more pathological mark. Key brain regions like the hippocampus and amygdala show altered epigenetic profiles after trauma.
So the experience is literally written onto the regulatory machinery of the brain cells.
In a very real sense, yes. The environment can leave a long-term script on how that stress response system operates.
And connecting the brain to our daily habits, there's a huge link now between epigenetics and sleep.
An absolutely essential one. Our internal 24-hour clock is run by genes like CLOCK and BMLO1, and they are deeply integrated with the epigenetic machinery.
How do they interact?
Well, the CLOCK protein itself is an enzyme. It's a histone acetyltransferase, a HAT.
So the clock protein itself is actively opening up chromatin.
In a rhythmic 24-hour cycle, it adds those on marks, these acetyl groups, to drive the expression of thousands of genes at the right time of day. This means your entire epigenome has a rhythm.
DNA accessibility is literally cycling with the sun.
Exactly. This ensures that repair processes ramp up at night and metabolic processes peak during the day.
And if you disrupt that rhythm with, say, shift work or chronic sleep deprivation,
you scramble the whole system, you disrupt that rhythmic gene expression, you alter the chromatin structure, and importantly, you accelerate the accumulation of epigenetic aging signals.
So sleep isn't just rest. It's a mandatory period of epigenetic maintenance.
It's biological housekeeping. At a neurological level, it's when you consolidate memories, which involves stabilizing those new neural connections through epigenetic writing.
And it's also when the brain physically cleans itself.
Right, through the glymphatic system. This waste disposal system becomes much more active during sleep, flushing out metabolic byproducts. And this is linked to epigenetics because that waste creates inflammatory stress.
And that stress can damage the chromatin and disrupt the machinery.
So getting enough quality sleep helps minimize the damage that forces the cell to engage in those disruptive, error-prone repair processes in the first place. It supports long-term regulatory fidelity.
Okay, so if the healthy epigenome is all about stability, then aging looks like the slow failure of that stability, a process called epigenetic drift.
That is the central insight. The aging epigenome has predictable features. Globally, you see a loss of DNA methylation. The silencers get weak. At the same time, you see specific gains of methylation, often shutting down genes you actually need.
But you've said the most important feature is the increase in noise.
That's the real hallmark. Instead of all your liver cells having the same precise epigenetic pattern, the patterns start to diverge. They become more variable, cell to cell. That coordination breaks down.
And this loss of coordination is the basis of the information loss theory of aging. What does that mean at a molecular level?
It means aging isn't just about accumulating random damage. It's the active, systematic degradation of the instructions for how to run the cell. The genetic information, the DNA is still there, but the regulatory information, the context, the control is being eroded.
And a culprit, ironically, is the very process of DNA repair.
It's a cruel biological trap. Your cells suffer thousands of DNA damage events every day. They have to fix them. But every time the repair machinery is called in, it has to temporarily disrupt the local epigenetic structure to get access to the break.
It has to move the histones out of the way.
Exactly. And then after the repair, it's supposed to put everything back perfectly. But this process is incredibly resource intensive. And there's a key vulnerability. The enzymes that are first responders to DNA breaks, the PARP enzymes, consume a vital molecule called NAD plus O. They burn through it.
And NAD plus is also needed for epigenetic maintenance.
Yes. A critical family of epigenetic regulators called sirtuins. They are essential for maintaining stable, silent chromatin. They run on NAD plus TOC. They need it as fuel.
So here's the trap. DNA damage activates PRP, which drains the NAD plus pool. That starves the sirtuins, so they can't do their job of maintaining the epigenetic code.
That's it. This endless cycle of damage and repair slowly degrades the regulatory structure. The information gets lost bit by bit over a lifetime. The management software gets corrupted by a million tiny emergency patches.
And this understanding led to one of the most exciting tools in the field, the epigenetic clocks.
They are incredibly powerful. These are basically algorithms that look at the methylation patterns at hundreds of specific sites across your genome and use that to estimate your biological age.
And that can be very different from your chronological age.
Dramatically different. The breakthrough was Steve Horvath's 2013 clock, which showed this astonishingly accurate correlation with age across almost all tissues. It proved that the decay of the epigenome is mathematically readable.
And the clocks have gotten much more sophisticated since then.
Oh, yes. The first-generation clocks were great at tracking time. The newer clocks, like Grimmage, are much better at predicting mortality risk and measuring health span.
How does it do that?
They focus on CPG sites that are linked to things we know are bad for us, like chronic inflammation or insulin resistance. They're weighted towards markers of immune system age and disease. So groomage is a better predictor of your time until death than your actual birthday is.
So if my biological age comes back as 10 years older than my chronological age?
That's the interpretation. It means you've accumulated epigenetic signatures associated with worse health outcomes. But we have to be careful. They are biomarkers of a state, not the full causal mechanism of aging itself. They tell you what is happening, but not precisely why.
But the link between this regulatory breakdown and disease is undeniable.
It's fundamental. Cancer, for example, is a disease of massive epigenetic failure. You see tumor suppressor genes get silenced by methylation and just chaotic histone patterns everywhere. The cell forgets it's a differentiated cell.
And we actually have drugs that target this.
We do. Some cancer therapies are epigenetic drugs that try to force the cancer cells to remember their identity and stop proliferating.
And for neurological disorders?
Neurons live a long time, so their epigenetic maintenance is critical. In diseases like Alzheimer's, we see clear signs of dysregulation and instability in the neuronal epigenome.
And then there's the whole world of metabolic disease.
Right. Obesity, diabetes, heart disease, they all have distinct epigenetic signatures. It's the perfect example of the environment-gene interface. A lifetime of diet and inflammation writes a disease history onto your regulatory software.
Which brings us to the most radical idea of all. If this is all just software, can you reverse it?
For centuries, the answer was no. Differentiation was a one-way street. Once the ball was in the valley, it was stuck.
Until cellular reprogramming.
Right.
The idea that you could push the ball back up the hill.
Right. If identity is actively maintained by this dynamic system, maybe you could forcibly rewind it. And the first hint of this came way back in 1962 with John Gurdon's frog experiments.
What did he do?
He took the nucleus from a specialized frog cell, like a skin cell, and put it into a frog egg that had its own nucleus removed. And sometimes that egg developed into a normal tadpole.
Wow, so the skin cell's DNA still had all the information to create a whole new animal.
It proved that identity wasn't from losing genes. It was imposed by the regulatory state. Identity was, in principle, reversible.
But the real revolution came in 2006 with Shinya Yamanaka.
A seismic discovery. Yamanaka showed you didn't need a mysterious egg cell. You could take a regular adult cell, like a skin fibroblast, introduce just 4 specific genes, OCT4 SOX2, KLF4, and c-MYC, or OSKM for short.
And you could turn it back into a stem cell, an induced pluripotent stem cell, or iPSC.
Exactly. These 4 factors are master regulators of pluripotency. They go in and systematically erase the cell's adult identity. They erase the epigenetic marks of aging. They effectively reset the cellular clock all the way back to 0.
That sounds like the fountain of youth. Why not just do that to an entire person?
Because a full reset is incredibly dangerous. When you completely erase a cell's identity, it loses control and just starts proliferating wildly. It forms tumors called teratomas, which are these chaotic messes of different tissue types.
So in a living animal, that would be lethal.
Absolutely. Continuous expression of these OSKM factors is a recipe for aggressive cancer, largely because one of them, c-MYC, is a powerful oncogene. Full-body reprogramming is not a viable path.
So the goal had to shift from a full reset to something more subtle, partial reprogramming.
Exactly. The new goal is precision. Can we transiently or cyclically express these factors just long enough to roll back some of the aging markers to reduce the noise without completely erasing the cell's essential identity?
To get the benefits without the catastrophic collapse.
It's about restoring youthful function, not obliterating identity. And we've seen this work in animal models. In rapidly aging mice, continuous activation of the factors made them sick.
But partial activation.
Cyclic activation on for 2 days, off for 5, led to improved tissue health and extended their lifespan. It's all about the dose and the timing.
That's a razor's edge between therapy and destruction.
It is. More recent work has used just 3 of the factors, OSK, leaving out the dangerous c-MYC, and has shown it can regenerate the optic nerve in old mice. Their cells got epigenetically younger while remaining neurons.
So the aging signature really is recoverable, at least in part.
It suggests we can access those youthful regulatory patterns without triggering a total identity collapse. But the constraints are immense.
It's not just about chemical tags, it's about the physical 3D structure of the genome.
Absolutely. The DNA isn't just a string. It's folded into specific neighborhoods called topologically associating domains, or TADEs.
These are like regulatory silos.
Yes, they ensure that the regulatory elements for one gene don't accidentally turn on a neighboring gene. They prevent crosstalk.
And with aging, this organization starts to break down.
The loops weaken, the boundaries blur, and you get more transcriptional noise. It's another layer of information loss.
Which explains why trying to reverse this is so risky. What are the biggest failure modes?
The first is identity collapse. You try to make a muscle cell younger, but instead it gets confused. It starts expressing genes for fat or scar tissue, and you end up with fibrosis instead of repair.
And then there's always the risk of cancer.
Always. The pathways for rejuvenation and the pathways for cancer are deeply intertwined. They both involve growth and proliferation. The biggest challenge is separating controlled therapeutic repair from uncontrolled malignant growth.
And even if you get the individual cell right, there's the whole tissue to consider.
A huge constraint. An organ's function depends on the coordination of millions of cells and their physical matrix. You can't just rejuvenate one cell in isolation without considering the whole system.
This is why the term reset is so misleading.
It is. Scientists prefer terms like recalibration or partial restoration. It's not a hard reset. It's a careful adjustment of the software.
And there are massive technical hurdles, just in terms of delivery.
How do you get these factors to only the liver at the right dose for just the right amount of time without causing a huge immune reaction? It's a massive engineering challenge.
And this is a field that's also prone to a lot of public hype and misunderstanding.
We have to be clear. Epigenetics doesn't mean your thoughts can magically control your genes. The effects are mechanistic, constrained, and probabilistic. We have to avoid the myths.
So looking ahead, what's a realistic timeline for all of this?
In the near term, say 5 to 10 years, I think we'll see epigenetic clocks become common clinical tools to track biological age. And we'll see more refined epigenetic drugs for cancer and inflammation. But reprogramming will remain highly experimental.
So the long-term goal isn't really about radical life extension.
The realistic goal is shifting toward epigenetic maintenance, slowing the rate of information loss to extend health span, the number of healthy, functional years of life.
And if we succeed, that immediately raises huge ethical questions.
It does. Who gets access to these therapies? What's the line between treatment and enhancement? We're dealing with a finely balanced system, and the potential for unintended, irreversible consequences is very real. We need precision over power. You know, the biggest insight from all of this might not be the fountain of youth. It might be the recognition that our biological time isn't fixed by our DNA sequence. It's dictated by the dynamic, complex, and surprisingly fragile organization of its regulatory managers. Aging isn't just damage, it's a loss of coordinated biological information.
If you enjoy these late-night explorations, subscribe to Brain Binge. It helps the channel grow. And if there's a topic you'd like us to explore next, leave a comment.